Okay, again, welcome everybody. Thank you for attending the second ALLODD webinar in the ALLODD webinar series. And today I will present our work together with Alexis Hadjigoula. So this is Alexis' PhD thesis essentially. And apologies for the long title, but this presentation has essentially two parts. The first part is the prediction of protein membrane interfaces using ensemble machine learning. And the second part is how we apply this prediction of protein membrane interfaces into the application to drugging these protein membrane interfaces. So my lab works a lot, has a long-standing interest in protein membrane interfaces in the context of peripheral membrane proteins. So you see here PI3K bound on a model cell membrane in ion channels, and you see here free energy perturbation cycles to predict the difference in free energy of binding in ligands that bind membrane proteins, in GPCRs, and of course also on pure membrane systems, from force field creation to atomistic simulations and coarse grain simulations. Also we're working with nanoparticle design for efficient and non-toxic membrane penetration. So targeting protein membrane interfaces, why is this important and why we work on it? So basically abnormal protein membrane attachment is involved in deregulated cellular pathways and in disease. And because the protein membrane binding is very important in disease, we want to reconsider the neglected accessibility of protein membrane interfaces by small molecules, because as a protein approaches the membrane and in deregulated pathways, the signaling can become aberrant and we may get association with diseases. So what if there was a way that we could modulate this protein membrane binding with small molecules? And this is what we want to do in this project. We basically want to tackle the complexity of the protein membrane interface. Also, the protein membrane interface is usually unknown. So we want to work on identifying exactly where peripheral membrane proteins bind. And we want to design computational workflows to study protein membrane interfaces, because while they exist partly, they don't exist as a coherent methodology. Also, the sites where a ligand may bind on these protein membrane interfaces are largely unknown. We don't have so much structural information for this interface. And we want to create a novel computational workflow for drugging these protein membrane interfaces and treat it as a novel drug design strategy. So the computational workflow for achieving this drug design strategy will start with the identification of where a membrane protein will bind. So the membrane binding domain identification of the protein. So this is the first part of the talk. The next step is basically to find the conformational ensembles of this protein and identify common binding pockets that are located at the membrane binding interface. But finding binding pockets is not always enough. It's not enough to have a binding pocket that you can work with and bind a small molecule. This pocket needs to be allosterically connected with functional modes of the protein or with the active site. And if it's allosterically connected and it has an effect on the function of the protein, then we consider this site to be druggable for allosteric ligands and on protein membrane interfaces. After identifying these druggable sites, we can then proceed with a normal virtual screening workflow of docking and scoring, compound selection, and in vivo and in vitro assays. So essentially what we want to do in this project, we want to answer three questions. First is, where is the membrane protein binding onto the cell membrane? The second question is there any druggable pocket close to the membrane site domain that we could potentially target small molecules? And the third question is this pocket that we found, is it allosterically connected with the active site or any other functional domain of the protein? And this is, of course, the relation to our grant, Allostery in drug discovery. So I will start with the first part, with the first question that we want to answer here, which is, where do peripheral membrane proteins bind onto the membrane? So we have a machine learning strategy for this, and the first thing in the machine learning is to have a hypothesis. And here the hypothesis is whether we can predict, or that we can predict the membrane penetrating amino acids. And for this we have to build a data set. We need to apply machine learning algorithm. We need an initial data set. And as data set, we use peripheral membrane proteins with known three-dimensional structures and experimentally known membrane penetrating amino acids, so that we create a binary classification problem, which is to distinguish amino acids that bind to the membrane and amino acids that don't bind to the membrane. Then, in order for our algorithm to predict accurately the membrane binding regions, we need to extract features, physicochemical and biochemical amino acid features, which will differentiate between membrane penetrating and non-penetrating amino acids. And then we will train supervised machine learning algorithms based on these features in order to finally distinguish, given an unknown protein, to distinguish whether this protein has penetrating amino acids to the membrane or non-penetrating, and where they are. And finally, to evaluate, we train the supervised algorithm and evaluate them in an independent test set. So our data set started by the known three-dimensional structures of 54 peripheral membrane proteins with experimentally known membrane penetrating amino acids. And we then split the data set in training and validation test sets. And of course, before we do that, we have to prepare all the data set, meaning to prepare all the proteins. And maybe I will continue here. So we split the data set into a validation set and a training set, and continue with data selections in the training set. And because only the amino acids in the protein interface can interact with the membrane, we dropped those with amino acids with residue depth smaller than 2.5 angstroms. And additionally, only hydrophobic amino acids insert to the membrane. So we removed non-hydrophobic amino acids. Then we applied the same data selection as shown here into our validation set. And furthermore, we assembled an independent test set of 11 peripheral membrane proteins with known three-dimensional structures and experimentally known membrane penetrating regions. And to ensure that the predictions are unbiased, we made sure that the proteins that we're using, the percentage of identical amino acids by all sequence pairs of all proteins in our dataset was below 40%. And so the validation and test sets can be considered independent or unbiased. So basically, the first step in the machine learning process is to collect your data and then to split them in a validation and training set where the whole training is happening and an independent test set. After that, you have to select, you have to calculate which features are important for the problem that you're studying. So the feature importance here was calculating using different ensemble tree-based methods. So basically, to calculate the features, you don't do it by hand, of course. We have an algorithm that will calculate based on the known data, which features are predicted to be very important for membrane binding. So we use the extreme randomized tree classifier as well as light GPM. And by measuring the number of times each feature was used to split the data across all ensemble of trees, we could validate whether a feature is important or not. And then we removed the features that were in at least 1% of feature importance in both methods. And what this gave us was the most important features from these tree-based algorithms. So the ones that were found to be most important are hydrophobicity, of course, it makes sense, solvent accessibility, secondary structures, but also apart from these, which were kind of evident, we also found some novel physicochemical insights for the mechanism with which peripheral membrane proteins interact with the membrane, because we found that important features are also the evolutionary conservation score, flexibility, and also the dihedral angles, how these are changing. So as mentioned, our dataset consists of 54 peripheral membrane proteins with experimentally known penetrating residues. And these were retrieved from literature search. And these were split into classes. So the membrane penetrating class and the non-penetrating class. And the first issue that we were faced with was that with the membrane penetrating amino acids are way less than non-penetrating, because of course you have limited data for peripheral membrane proteins, while for soluble proteins, you have so many proteins that are soluble. So you have much less membrane penetrating amino acids than non-penetrating. So basically the non-penetrating has a lot of points here, and the penetrating not so much. So this leads to an imbalanced class problem that we have to deal with. And to solve this issue, one can utilize something that's called over and under sampling. So these are two techniques. There's also a third technique, which is called weights. So basically here we use the three techniques. The first one is to use weights on the samples, where more emphasis is given by the classifiers in the minority sample. And then the second one is to oversample the minority class. The minority class is the penetrating amino acids. And the third technique is to undersample the majority class with sample selection methods. So in the end, this results to a balanced training set with these techniques. And the initial training set, which is all these amino acids that I just explained, we split them in 5 different pieces, which we called folds. So we split them in 5 folds in order to later perform optimization of the parameter space where we're going to use our machine learning algorithm using 5-fold cross-validation. We also used class balancing in each of the folds using the techniques that I mentioned before. The different over and under sampling. And the reason for applying class balancing in these 5 folds separately is to prevent information leak in the validation fold during the 5-fold cross-validation procedure. In the end, this gave us 6 different training sets split in these 5 folds, which were used in 5-fold cross-validation. So except from the features and the data set, the success of the machine learning is also based on the algorithms that we're going to use. So here we basically used a lot of different algorithms to train our data set, because these classifiers that will select which amino acids are going to be penetrating and non-penetrating, they work with different methods, and essentially some have the potential to predict the correct answer and some not. But the problem is that we don't know which one is which. So we have to choose wisely. And basically what we did here was to repeat all this training for 21 different classifiers. And for every classifier and parameter combination, we trained it 5 times. And basically, in the end, we ended up by training and validating 21 machine learning classifiers. And this resulted also to performing hyperparameter optimization using the randomized search cross-validation technique in a wide range of hyperparameter space. Essentially then training these 21 classifiers and using 5-fold cross-validation and using different parameters. This results in hundreds of thousands of models. And in the end, we basically have to choose which is the classifier, the algorithm that best predicts the penetrating amino acids more correctly. And how we can know? We can basically use something that is called the Meta-classifier, which is a voting algorithm that will tell us which of the 21 classifiers, and of course, all the different parameters, will have the best predictive power. And so we basically then kept the best model for each classifier that has the best F1 score with respect to predicting the membrane penetrating versus the non-penetrating amino acids, according always to the 5-fold cross validation. So this voting classifier, as I mentioned, essentially gives you a majority voting from these 21 classifiers. And we also use the stacking Meta-classifier, where we tried all possible combinations of the first level classifiers. So we also took, okay, not to be confusing, I just will mention this step. So basically, in the end, the best Meta-classifier model was a voting classifier of 5 first-level classifiers, which achieved an 86% of F1 score. And yes, it's right here, 86% of F1 score, and an MCC of 0.71 in classifying penetrating from non-penetrating amino acids. And these are the 5 classifiers, because I told you in the beginning that we use 21 algorithms, but actually the voting classifier, so this Meta-classifier and the stacking classifier, told us that these 5 algorithms that are called classifiers are the best ones that we have to choose for our specific problem. So using all this, let's say, complicated methodology, in the end, we inserted the validation set, which doesn't have, which is not known to the algorithm, to check whether our predictions were accurate. So, so basically, we can see our model predictions in the proteins of the validation set. With red, we see the true positives. So with red, we have true positives. With green, we have false negatives. And with purple, false positives. So the putative membrane plane is depicted with a red dotted line. So again, I will say it, you can see it color-coded here. True positives are experimentally known membrane penetrating amino acids predicted by our tool. False negatives are known amino acids that our algorithm did not predict. And false positive is amino acids predicted from our tool that have not been experimentally proven. But of course, one can see that in this case, some, let's say, false positive is actually could be a true positive, but it's just not assessed by experimental methods. So it's a little bit tricky, what you call false negative and false positive, because here this false positive means that it's not assessed by experimental methods, but in fact, it is a true positive. While in this case, this amino acid is not on the surface of the membrane. So this is a true false positive. Right. So basically, if we consider these predictions, we see that the algorithm performs quite well. And considering that these predicted membrane penetrating amino acids are in fact located in the plane of the protein membrane interface, and they can consider true positives, like I told you here, then we reassess the scores, and the score from 86% can become 92% if you take into account these predictions. And the MCC can go from 0.71 to 0.84. Yeah, because you can see here that while this is classified as false positive because there's no experimental evidence, in fact, it is located at the protein membrane interface that we know. So another thing for the protein membrane interface, was to compare the protein membrane interface prediction of our ensemble classifier model with those of other computational tools, such as PPM and MODA, to other programs. So we applied our ensemble model to the proteins of the validation set, this time retaining all amino acids. And our results showed that several non-hydrophobic amino acids were predicted as membrane penetrating, both in the actual protein membrane interface, but also in the solvent exposed regions of the protein. To restrict such false positive predictions, we incorporated in the algorithm the condition that non-hydrophobic amino acids in a distance of more than 14 angstroms from at least one of the predicted hydrophobic amino acids are discarded. And again, this red dotted line is the membrane plane. So when we compare with PPM and MODA, these two other programs, our ensemble model displays equal performance or better in some cases in the validation set than PPM or MODA. And in the retinoid isomerohydrolase example, PPM, for example, completely falsely predicts the membrane plane. And MODA correctly places the plane, but it has false positive predictions, while our model seemed to be more robust. So in addition to that, we had an independent test set, as I said in the beginning. So we tested the performance of our tool in this independent test set with known membrane penetrating regions. And here you can see that our tool predicts almost always this, it predicts always the protein membrane penetrating regions, but in this case, it also has here some false positive predictions. But in fact, when I show you these false positive predictions, when we examined it further, we found that with further manual inspection of these false positive predictions, those that we thought are false positive, are actually near N or C termini or near missing loops, or actually at the area where a protein is interacting with another protein, so at protein membrane, protein-protein interfaces. So basically, what we are finding is that here you see a false positive prediction, but in fact, it's not a false positive prediction because basically this is found right here where the protein is interacting with another dimer. So this dimer forms a tetramer, and the dimer of dimers has this false positive prediction right at the interface. So basically, with this algorithm, you can not only find the protein membrane penetrating amino acids and regions, but also possibly you could identify protein-protein interactions because protein membrane interactions are similar to protein-protein interactions. So in the end, we tested also the performance of this tool because I mentioned I'm talking all this time about peripheral membrane proteins, but in fact, we wanted to test it for transmembrane proteins because we know that transmembrane proteins are a big class of drug targets. And we wanted to test the performance of our tool in 9 transmembrane enzymes, which include a soluble domain as well as a transmembrane domain. And the membrane penetrating amino acids predicted by a tool are depicted with red here, and the plane of the membrane is with red dotted lines. And in agreement with experimental results, our model correctly predicts amino acids that line the hydrophobic lipid bilayer core. See here, for example, or here. And of course, more testing is required for us to conclude that our ensemble classifier model is competent for the prediction of membrane interacting amino acids of transmembrane proteins, but the results look already good. So after concluding with this algorithm, we wanted to also create a workflow for targeting protein membrane interfaces with small molecules. So apart from predicting protein membrane interface, we wanted to create a workflow for predicting binding sites in protein conformational ensembles, and doing so by the implementation of a web server that would implement this workflow, which we call DREAMM. And in DREAMM, first we predict the protein membrane interfaces. Then we apply our protocol to a conformational ensemble selection to predict pockets. And then on these pockets, we can perform JAK design. So basically, to predict protein membrane interfaces, as mentioned, we use our designed novel ensemble machine learning classifier or model for predicting membrane penetrating amino acids. Considering the dynamic nature of the proteins, we apply also an algorithm from the literature that is called expose. So the user can use their own conformational ensemble, but in case conformational ensemble is not available, with expose, one can generate a protein conformational ensemble without performing molecular dynamics simulations. And finally, to predict binding pockets in protein conformational ensembles, we facilitate the step by implementing the open source binding site detection software P2Rank. So we apply P2Rank in all the conformation that either the user inputs or expose generates. We keep binding sites at a distance of 5 angstroms from the predicted protein membrane penetrating amino acids. And then we create a publication-ready visualization for the end user, as you can see here. And of course, we also give all the results available as files to download. To assist the interpretation of results, because you may have 10 or up to 50 conformations, what we do with the binding pockets is that we cluster them with k-means. And one of the main drawback of k-means, as we know, is that we need to define the number of clusters a priori. And because we don't want to do this manually and to choose the correct number of clusters that we want to find, we use the silhouette score, which basically displays a measure of how close each point is with the points of other clusters, minus the distance from the points in the same cluster. And this basically defines or converges to a final cluster score with the optimal K. So basically you see here that after 10, essentially the silhouette score does not increase. So 10 should be your preferred cluster number. And after clustering the sites, the binding sites, we provide binding site statistics to the user, such as the number of conformation, where the binding sites are apparent, and the mean P2Rank score, and others. This computational workflow has been, for dragging protein membrane interfaces, has been implemented as a web server, which can be accessed and used by this link. And an important attribute of the web server is that it has a queuing system. It allows you to run simultaneously with other users, and a unique URL is provided to you, which can be accessed and bookmarked for later use. And now let's see, and I forgot to mention that it was onboarded in the European Open Science Cloud, so it is accessible to all for use. And now let's see a video tutorial about exactly how the DREAMM web server works. So this is our website. So initially, the user has to choose whether they want to search for binding pockets near the protein membrane interfaces, because maybe some of the users don't want to use a full workflow, but just want to predict protein membrane interfaces. So the first step is just to choose. So they can do this by uploading a PDB file or by selecting the 3-letter PDB code. Yeah, so here it is selected. Yeah, some more details about the chains. There's also a manual that you can use. So if, again, you don't have an NMR structure with a conformational ensemble, or you don't have a molecular dynamics trajectory, then you can choose to generate a protein conformational ensemble using expose. And for that, we can try an example. P10 protein. So now the first step is to predict the protein membrane interfaces. So the machine learning algorithm is running in the background. Then the page is reloading. But basically, if you bookmark this URL that is right here, then you can come back at a later time to check. Okay, let's see the results. So this is the first step of predicting where the protein binds onto the membrane. So we display that with J small, and we see that apparently the membrane plane is formed by the line connecting these groups of amino acids. And the results for this first part, which is the prediction of the protein membrane interfaces, just download the CSV file with the amino acids that are located. Of course, you can also see it visually, but it's useful also to have the text. So if we want to search for binding pockets, now close to that, we can, yes, we can upload or give a PDB code in this case, and ask Expose to generate 20 conformations. So because you are giving the algorithm 20 different conformations and it has to look, it takes approximately half an hour. Let's see the results. Okay, so these are the membrane penetrating amino acids. And now the next step is to also check for the okay, this is just for the protein membrane prediction, but now you will also get the different conformations from Expose that is being output to you together with pockets where you have P2 rank predicting binding pockets, different binding pockets. And we have created Biomol files, which you can open, and you can visualize these pockets, and only the pockets at a distance of 5 angstroms from membrane penetrating amino acids will be displayed. If you wanted to have other regions of the protein, you can just use P2 rank directly. Here we use this tool because we want to find pockets at the protein membrane interface. So the results, yes, the results are also shown as TXT files. And also the algorithm will tell you whether like these binding pockets are found in all the conformations or they're found in some conformations. That's also important. I don't know if I missed anything else. Right, so these are just details about where these pockets are and so on. So to continue, of course, we found these pockets, but the main question especially for the design of allosteric ligands that target the protein membrane interfaces, whether these sites are allosteric and whether these sites are connecting their motion to the active site or to other functional domains of the protein. So basically, allostery can be defined as the functional change of a protein generated by perturbation from the binding of an effector at a non-orthosteric site, such as the protein membrane interface. And this is called an allosteric site. And it is proposed that the allosteric information travels in the protein through pre-existing allosteric networks. And there are methods that have been already created to define these pathways or to describe these pathways and to provide them, computational methods. And we have tested actually some of these methods, and these methods actually work quite well. So here, for example, we have used dynamical network analysis to identify the allosteric networks between the membrane binding domain and functional domains of the protein. And here, for example, we show that PI3K, which is attaching on the cell membrane, has a mutation, which is called the histidine 1047 arginine mutation, that actually affects the way with which this protein is attaching on the membrane. And we wanted to understand how this mutation, here's the wild type, is connected to the membrane binding loop. And we performed the dynamical network analysis and found that basically when you have the mutation, the arginine, the path connecting the mutation, the mutant residue with a membrane binding loop is much shorter. So these residues communicate rather than the wild type. So this is basically building cross correlations between amino acids, motional cross correlations. Next, we tried the dynamical cross-correlation based methods that are shown to be effective also in capturing the allosteric networks in MD simulations. But we know that MD simulations are computationally expensive and time-consuming. So to resolve these problems, we wanted to understand whether we can train graph neural networks to predict this allosteric cross-linking of amino acids. And one example of that is if you can relate, there's an analogy between traffic and allosteric pathways in protein. So DeepMind uses graph neural networks to enhance Google Maps estimated time of arrival accuracy by 50%. So basically they use graph neural networks to predict the traffic on the street. And here we could predict the dynamics of the protein inside the protein, where different parts of the protein communicate with each other. So we could also think that we can use graph neural networks to predict the traffic within the protein. So the plan here in this part of Allostery drug discovery project is to train graph neural networks to predict allosteric cross-linking of amino acids. And we assume that dynamic communication pathways are at least partially conserved between the proteins. And if this is true, supervised machine learning methods should be able to identify common patterns in these communication networks and predict dynamic information for novel proteins. So we want to transform proteins into graphs and input them into neural networks in order to predict the amino acid cross correlations. So basically, this is the plan for Allostery drug discovery. So I want to use machine learning in order to predict communication network pathways. And with that, I would like to finish. I would like to thank Alexis for doing this wonderful project in this lab, for building this DREAMM web server, and for all the great collaboration that we had. And of course, I would like to thank Allostery drug discovery and all of you for being here with us today. And with that, I think I could stop here and take questions. Thank you, Marco. I see Marco is clapping.
Thank you, Zoe. Maybe I can just to break the ice.
Yes, thank you.
It's a very general question. There's a question I have as a non-expert in machine learning. We now see all these different methods for machine learning popping up, and everywhere it's being used. Could you comment on the learning curve for those methods?
Yes. I will. So actually, I was also, when this whole hype, the machine learning hype started, I was also felt a little bit out of my waters, because this is, you know, like a new buzzword and no one really knows what it means. But when I started working with Alexis on the project and reading the literature and slowly getting into it, I understood that this is not a new methodology. This is a very old, it's 50 years old that it has started to being used. For example, linear regression is a machine learning methodology. So these methods existed before. It's just that the data was not there that we could use. There was not enough data. So machine learning uses a lot of data. And because before we didn't have so much data, we couldn't use it. Now with genomics, and with crystal structures becoming widely accessible, we have data that we can use. So to answer the question about the learning curve, I think once a person wants to get used to it and starts reading the basics, they realize that it's nothing unknown. It's old methods with, of course, newer technologies, because Google is bringing new algorithms. But the methods behind these algorithms are pretty straightforward. So I would say that to start with machine learning, one would understand the theory, definitely then learn the basics of Python, then data frames with Python, and then some tutorials with machine learning online we can find in Coursera or other free courses. And it's quite straightforward. I think in 1 month, a person could really understand and get used to it. But it's a great question, I think. But maybe you could also add something, because I know that you're also working a little bit on that.
Well, I'm sort of starting to test the waters. So I still haven't answered that question. I hope it is as you say.
Okay, yeah, you know, it is true, because also I'm no more expert than you or than anybody in the audience. We just really went with Alexis, who of course, he has finished, he's a computer scientist, and he's the one that could program, but he also could guide me through the steps. And I just realized that it's not something that is not possible to learn. I see hand from Sonia.
Yes, thank you very much for the great talk. I just wanted to know, are you continuing on improving your machine learning algorithm now, like that you know that you could also work with transmembrane proteins, so that you would like include more, like, yeah?
Yes, we have, indeed, we have a lot of ideas, and we have a lot of things that we want to test. We have more data. We recently discussed with Natalie Reuter, that is also working on the same problem and has a bigger database. As you saw, our data set was only 54 proteins. Of course, the 54 proteins translate to many more amino acids, but still, it's a small data set. And Natalie has a bigger data set. So we are working on improving the algorithm. Also, maybe considering other descriptors, such as the curvature of the membrane or other physicochemical properties that could be useful. And yes, we're very interested in this problem because it has been an interface that is not very much worked with because of the complexity of this interface. So we're continuously working on improving it. And the last part with the graph neural networks and how we could predict the allosteric networks using machine learning is going to be Bruno's project. So let's see.
I almost thought so.
Yes.
Okay, great to hear that it's ongoing.
Yes, thank you. Thank you, Sonia. Marco.
Yes, thank you for the interesting talk. I have one question related to basically the last part of your talk. So you were talking about applying machine learning to identify allosteric pathways, communications between residues and such. And you just mentioned answering to Xavi that what is very important is data. So you have to train a model anyway. So what kind of data would you use to validate your model? Because I don't know whether there's experimental information about allosteric pathways.
That's right. Great question. And actually what we are doing, our training set will be 500 molecular dynamic simulations. So we basically, what we want to do with this algorithm is to avoid having to do molecular dynamic simulations. So if we have enough data from molecular dynamics, and we saw that we showed that we could predict allosteric networks with or the information that we have, as you said, it's not so great. We could predict them with dynamical network analysis or with another method by Greg Bauman cards. And the goal here is, and it was not clear, so thank you for the question, if we can avoid running molecular dynamics and directly in seconds to predict the allosteric networks as they would be predicted by molecular dynamics. That would be the closest that we could get to reality.
Yeah, and I wanted you to comment on a related question. So you talked about a server, I think, that I was not aware of. So is XProZ, or is a method for producing
it's a software.
It's a software, yeah. So is it also based on machine learning? Is it one of these methodologies that use a neural network to produce?
Yes, that's correct. That's a machine learning based method for, no, that's P2Rank. Sorry, that's P2Rank. Okay. And this one, XProZ, is a geometry based method. So it's, I think, maybe linked to what you're saying. Yeah, but I don't remember the details of that.
And in your experience, or in your hands, how good are these algorithms? So do they produce really meaningful configurations, and how much more efficient are they relative to molecular dynamics?
Yeah, that's a good question. I mean, we read the paper, the data looked good, and we didn't try it. So I don't know how I could assess it. I mean, the data look good in the paper, but I don't know how we could assess the different conformational ensembles that are being generated, and actually didn't check the RMSD that they produce. So that's something that we could do, I guess. Taking only the data from the paper can be misleading.
Well, it's always good.
It's always good. That's right, always skewed, but it's a good question.
Thank you very much.
Do we have another question? Okay, I thought Bruno would ask something, maybe.
Yeah, actually, I was trying to I have actually two questions.
Okay.
But I was thinking maybe asking you when you were here. I want to give the opportunity to the other attendees, but well, I will ask. In the first part of the methodology, you mentioned about the a 40%. You take out some proteins that have no more than 40% of identity between all the sequences, right?
That's right.
How did you decide this 40%?
The 40%, this is known in the literature. Above 40%, you have big identity between the proteins. This is a limit that is okay to be considered. Like below 40%, well, so it's between 20 and 40. Some would say even 22% would be identical, but we chose the 40%.
Okay,
that's from the literature, yeah.
Okay, and you mentioned that you did this step in order to unbias your methodology, right?
Yes, because if you have all the proteins are of the same protein family, then the training will be biased to this protein family.
Okay. Another question that I had is that I saw in your methodology that you mentioned about the computing of different descriptors for the proteins you have in your data set. Could you please talk to us about this? Which kind of descriptors were these? Maybe do you include one dimension, 2 dimension, 3 dimension descriptors? What are these descriptors about?
So the descriptors that came out, some of the descriptors were quite obvious. So the hydrophobicity of the amino acid was one important descriptor, and the solvent accessibility, which is kind of intuitive because you know that you're going to bind at the protein membrane interface. So you know that the hydrophobicity and solvent exposures are going to play a role. And also the secondary structure, whether it's a loop or it's an alpha helix and so on. And so these were kind of obvious, but then other physicochemical descriptors came out as important, such as the evolutionary conservation score. So if an amino acid is conserved across evolution, then it looks like this amino acid has a functional role. And this feature was important in our algorithm. Also, the flexibility and the dihedral angles, they also were proven to be an important feature. These are some examples.
Okay. And going further in this question, maybe we can go further in this. I imagine that the answer is yes. Maybe we can discuss it with Alexius in the future, but did you perform any kind of selection for these descriptors? Do you remember any methodology you followed to reduce the number of these descriptors?
Yes, so we removed the features that were in the last 1% of feature importance in both methods. So if it was in the bottom, we removed these features.
Okay. Okay, thank you so much. I have no more questions.
Okay.
Thank you so much.
Okay. Carles.
Yeah, hi.
Hello.
So thank you for the nice talk. So I have one curiosity. So with the graph neural networks, so beyond identifying the network of residues connecting two parts of the protein, right, which is one of the main objectives, if I understood well.
Yes.
So do you aim also to quantify how strong is this coupling, this dynamical cap? So in a way that you could maybe differentiate between a potential allosteric site and another binding site.
Yes, correct. And that's actually the reason why we were inspired by Google Maps, because they also quantify the traffic. And the traffic is an important factor. So if the traffic is enhanced, it may mean that it is a pathway that is more important than another one. So yes, we think that with this way, we could understand also and quantify which pathways are more populated or more traffic goes through, so they could be more significant. But I mean, we haven't started this yet, so it may be pure imagination or daydreaming. I don't know. We will see. I think there will be an outcome. But as mentioned earlier to Marco, the baseline will be molecular dynamic simulations that we perform on a known set of proteins. Another question that is interesting that we still are not sure how to answer is whether we have to create different training sets for different protein classes. For example, whether GPCRs have to be trained separately than kinases, than ion channels, ligand-gated ion channels, and so on. Or whether you could create a superset of all the classes, and then the algorithm could predict any class that you input. I don't know the answer to this question. I mean, my feeling is that we have to separate the classes, but I'm not sure. Maybe if you put everything together, it will work. I don't know yet. It will be another webinar in 3 years.
Okay, thank you.
Okay. If we don't have any other questions, it's 5 o'clock. 5 o'clock in Athens, 4 o'clock for you, most of you. And so with that, thank you so much for attending. The webinar will be uploading in our YouTube channel. And see you all very soon, at the latest in Barcelona. And for the students, you have a journal club that's coming up. So see you all in person in Barcelona. Looking forward.
Bye-bye. Thank you.
Thank you, Zoe.
Bye. Bye, everyone.
Bye.
Bye, Zoe.